morning. I'm extremely delighted to participate in today's webinar and to give a brief presentation of the More company. Here you can see an image of our current product. And as mentioned in the introduction, we work on a laboratory platform on the optimization of 3D cell cultures. Since we are the first speaking today, I'll bring along some context so that you kind of get an idea in which kind of industry we are moving, where we are active, and where the main bottlenecks are. We are active in drug development, and this is a process that had been going on unchanged for decades now. There are numerous statistics about it. And the current one, for example, by Peter, tells us that every year more than 190 million animals are used for various and different types of experiments. In oncology, it takes about 15 years for a drug to be placed on the market. And normally we are told this is worth it. We have made great progress in drug development. So the whole process is extremely inefficient still. Currently, less than 1 out of 20 compounds will finally be approved for marketing and provide and can turn into a successful therapy on the market. Well, we are not the only ones to tackle this problem. Luckily, there is a movement which helps us, that people say, can't you reshape this development process, make it more innovative so that we make it more efficient and let it have more success. And the US administration said so as well. It was the Environmental Agency which said as of 2035 we do no longer want to fund any animal experiments. So industry is looking for alternatives everywhere. One of the alternatives is an organoid. Here you can see it, a hard organoid on the right-hand side. It has the size of a sesame grain based on human cells, and it offers an alternative as to how to test drugs in a laboratory environment for safety. It offers various advantages. It's incorporated in the official FDA guidelines, so you can really do drug testing to put drug on the market, and it's made based on human cells. So a lot of studies go to show that the results on organoids as to how well a therapy will work can be transferred and translated on a human being. What this means for the pharmaceutical companies, it means they have a better databases making a drug more quickly available. We haven't invented the organoids, but we saw a problem with the future standardization. There are dozens, if not hundreds of organoids needing expertise. For example, in our team, we have a postdoc biologist which has been doing nothing but liver organoids for 8 or 9 years. Which organization can say, okay, we need toxicity for liver and brain and the heart. In terms of data, who can afford 3 postdocs working in the 3 different fields? Are there enough postdocs at all? And so we said, oh, we need to have a look at the whole plethora of organoids, this portfolio of organoids, which needs standardization and simplification. And here is our solution. Well, we as engineers, because we originally are engineers, had a look at the 3D cell culture process and thought about and how to improve it so that you don't have 8, 9, or 10 years of experience needed, but that lab technicians, for example, can apply this new technology in a very simple and reproducible way. All things look as follows. The protocols, or the kind of cooking recipes of organoids, are very specialized. Every research group has its own protocols and only relies on their own protocols, or perhaps the publications of a professor whom you know in person. The cell culture material materials, so the ingredients for organoids are selected, but this is based on experience. Oh, we have done that in the past like this, it works, and we don't want to change anything. Well, now we go to the lab, start to produce an organoid, a lot of manual pipetting that may last for weeks or months. So in order to have such an organoid grown, and it should have the right properties. You use the microscope or do any assays to characterize them appropriately. That's as the process looks today. But this is also a process requiring lots of expertise, and it's not transparent. We are going to change this. We at More offer a lab platform. We do not only have a robot in hardware that automates the experiments, but right at the outset, we have a software and a huge database of validated protocols, which means we bring in a lot of transparency into this process. And what does that mean? A user that never before worked with cardiac organoids before can enter our platform, log onto it, and get have a look at a protocol validated by us. It's a bit like HelloFresh. You have a recipe, and you get the ingredients as well. In the lab, you can quickly start to grow organoids. And we also offer an optimization of cell culture materials, which means in turn that we have a look at every individual growth factor that is included in this culture. So we have a look at this process, break it down in the individual parts to find the success driving factors so that we can offer better quality and a higher throughput in terms of culture. Our robot also comprises a quality assurance unit, which is integrated, which offers the researcher constant information so that we always know how well the cell culture runs. Is there an organoid that doesn't develop as expected, so that the researchers may interfere quickly and waste very little time on experiments that don't lead to a result. On a product level, it looks as follows. We set up a benchtop lab robot and everything around it. The special aspect is that we also have a biosafety level 2 hood. So it's possible without this hood to work with patient's tissue, because the patient-generated organoids directly from the tissue are the highest degree of personalization. We also use a special pipetting technology, which is very precise, but also very soft towards the cells, so that we have extremely good viability data on our organoids and are cultivated in our system. Typical applications that run on our robot is the cell seeding out of a little vial cell solution with 96-well format, an organ on a chip for more complex analyses. In addition, we have the automatic medium exchange, so that saves a lot of time, several hours per day for a researcher, which is saved of stupid work, by the way. We have very assays to characterize the organoids, but we also work on drug screening or toxicology screenings. All this can be done in an automated and transparent way. Where are we now? Currently, we have entered into several partnerships, and we have validated our prototype. We concentrated and focused very much on hardware together with industry. How can we make sure that these expensive samples, human cells and stem cells, that they are treated appropriately? And we did that in coordination with a lot of companies and got excellent data that our solution works better than the human hand. A lot of biotech and research institutes, we worked together and started the Organoids. Our value proposition is We do all the organoids in a simple way. So our biologist, who is a liver expert, also did cardiac organoids, brain or pancreas organoids, and liver organoids. And this is what she could do by using our platform with excellent results. We have higher cell viability compared to the manual process, but also time savings because this process has been automated so that our biologists could concentrate on the main focus of her work. And this is some data I brought along for you. We've all got it up on our website as application notes, or if you send me a message, I'll send it over to you as a PDF. These are the first few studies with the first few organoids with a high throughput. And one ability you can see on the left-hand side is the transportation of organoids, which is very meaningful for filling organoids on a chip, but also for imaging. So you don't have the same carrier, the same kind of plate as a basis, but you have to transport it. We can do it with 100% reliability without losing a single organoid or even damaging it. So we had to look at the cell culture process in detail, recognize the bottleneck, and with our automated solution, we were able to solve the problem. We are a young company, a young team. We are 8 people at the moment. We were set up in March 2023. And before we were in the X6 phase as a spin-off of the university. I'm a chemical engineer myself. I'm Lucas, and I have accompanied this project in academic research for 7 years. And then I got David into the boat, and we were also working with it. Now we have got Julia. She works with organoids and working with stem cells, and she is really the expert in hard science. We are a quickly growing team with a view to systems biology, where we have generated a lot of data on our platform we would like to analyze to improve our cell cultures. And for this purpose, we also need operational hands, which set up the robots and developed the robots. We have got finished our first seed funding round, 2.3 million, because our product, the platform you saw in one of the pictures, which will be placed on the market in as of January 2025. And we look for reinforcement people that come in and help us. We are located in the Spectrum building and center of Hamburg, the university. We closely cooperate with the university hospital. We have access to patients' tissues, for example. So you can look forward to a very innovative, very positive team and its environment, because we are very much looking forward to improve the status quo in organoid research. Last but not least, whom are we looking for? Scientists who will work together with Julia and have a PhD or postdoc level in stem cell research or organoid field. We also look for lab technicians that help us to conduct the orders and contracts we do it. Life science biology is very important. We want to get that. There are 2 more roles which are more customer facing, business development and application scientists besides bioinformatics. We want to be prepared to talk to our customers and to find cool projects in cooperation with the pharmaceutical industry in order to bring organoids to broad application. Thank you very much for your time, and I'm very much looking forward to the breakout room session. Please send me a message if you would like to, and if you would like to talk with us, send us your CV, please.
Ja, super, ganz lieben Dank erstmal.
Super, thank you very much to Lucas. That was extremely interesting. And I think there are some people who thought, oh, I would have loved to have this as well, or even today. And we've also got some questions.
Also, da hattest du erzählt, ihr habt Herz- und Leberorganoide,
Well,
Leber auch
you
deswegen,
have
weil
heart and
eure,
liver
Biologin
organoids,
da
you
auch
said, because your biologist is
Ähm,
specialized
nach
in these
welchen
organs.
Kriterien wählt ihr diese
According to
Organoide
which
aus? Weil ihr habt ja
criteria
relativ
are
viele
the
und irgendwie muss man ja
organoids
anfangen.
made and
Was ist
selected?
euer Fokus da? Worauf
What's your
achtet ihr?
focus here? What do you
Ja,
pay a
die
lot of
Marktslide
attention
habe
to?
ich jetzt nicht hier im Deck,
Well,
aber
I
das
haven't
ist
got the
genau
market
erklärt.
slide
Also wir
here
haben uns
on
eben
the
die
deck,
Top
but
3
I can
Anwendungen für
explain it. The
Organoids
top
angeguckt, die
3
es aktuell gibt.
applications
Das
for
Top 1 ist Drug
organoids.
Screening, Top
It's
2
first drug screening,
Toxicology,
second
Top 3
toxicology,
Disease
top
Modeling.
3 disease
Modeling haben wir ein
modeling.
bisschen ausgeklammert,
Disease
weil das
modeling
ein
that
sehr
we left
akademisches
aside.
Thema
It's a
oft
very
ist und
academic
weil das sehr
subject
schwer zu
quite often, and
standardisieren
it's
ist.
very
Und da
difficult
sind
to
wir dann
standardize it.
tatsächlich auch nach Gesprächen mit
And after
Pharmaunternehmen sehr
talking to
schnell auf den
pharmaceutical companies,
Toxicology-Bereich
we
gekommen. Das sind so ungefähr
concentrate and focused on
30% aller
toxicology.
Organoid-Anwendungen. Und ja, wo
The
macht man
area 30% of
Toxicology?
all
Das ist die
applications.
Leber,
Where do
das
you
ist
do
das Hirn und das ist das
toxicology?
Herz.
The brain, the
Das
heart,
heißt,
and the liver.
daher kamen unsere
And
ersten 3
hence,
Organoids. Und
this
die
is where our
Pancreas,
first 3
junges
organoids came
Startup, haben
from.
wir
And
recht opportunistisch
pancreas,
gemacht,
a young
weil wir da einen
startup,
coolen
we did it in opportunistic
Kollaborationspartner
way
haben,
because we
der
have a
uns
cool
eben auch direkt
cooperation partner
Patientengewebe oder
that makes
Zellen aus
patients' tissues
Patientengewebe
or cells from
zur
patients'
Verfügung gestellt
tissues
hat. Das
available
heißt, das war das
to
erste
us.
Organoid, was
So
nicht aus
this was the
Stammzellen
first
kam.
organoid,
Und
not
das war eben für uns auch so ein Proof
building
of
on
Concept, dass wir zeigen
stem
können, hey,
cells.
wir
So it
sind
was a
wirklich
proof
nicht
of
nur die
concept so that
Stammzellleute,
we can
sondern wir
show they are
kümmern uns
not
auch um
only the stem
Knochenzellen.
cell people here, but also commercial aspect.
Dann noch eine Frage und
Another
zwar, wie
question.
macht ihr euch bekannt an
How do you get
Universitäten
yourself known at
und wie
universities?
viele
How do you spread the
Grundlagenforschungsgruppen
word?
haben über
And how
morgen zum
many
ersten Mal
groups of
angefangen
fundamental
mit Organoiden
research
oder
have started to work with
3D-Bildkulturen zu
organoids or
arbeiten?
3D cell
Wie
cultures?
machen wir uns bekannt?
Well, how do
Ja,
we get
Webinars wie
ourselves known?
diese und
Through
diverse
webinars like
Konferenzen.
this
Also ich
one,
war zum Beispiel
conferences,
jetzt
for example,
dieses Jahr
I
auf den deutschen
attended this year the
Biotechnologietagen, wo wir
German
das vorgestellt haben.
biotechnology days
Oder
where we
eben auch in
presented
diversen
our product,
Startup-Programmen, die oft
or
an Unis
various
verbunden
startup
sind.
programs linked
Und
to various
ja, ich sag mal,
universities.
guter Kontakt
And to be
besteht
in
jetzt
good
rein
contact,
in
well,
ja, so ungefähr 10 akademische Gruppen.
good
Und
contact with
davon
10
würde ich sagen, hat die
academic
Hälfte noch
groups,
nie mit Organoids
half of which
gearbeitet.
have not yet worked with
Wirklich
any
von Grund
organoids
auf
so far.
etabliert haben wir bisher nur das
Well, we've only well
Cardiac-Organoid.
staffed the cardiac
Die anderen
organoid,
Organoids waren
which is well
tatsächlich alles mit
established. The
Partnern,
others
die
were
jetzt
developed
schon
together with
im Bereich
partners
aktiv sind und da
active in
Erfahrung
this
haben.
area or who have a lot of experience in this field.
Dann,
Well, next,
da hattest du ja gesagt, das ist so ein
you
Benchtop.
mentioned
Also
it's
kann
a
man sich
kind of
das so
bench
vorstellen, dass man das
top.
tatsächlich einfach irgendwo
Well, you can
draufsetzt und wie
say
viel
you
Platz
put
braucht
it
das?
Genau. Also der
on
passt
a
auf einen
bench, and
normalen
how much
Labortisch
place do
drauf.
you
Vielleicht ist da auch die
or
Abbildung nicht
space
so
do you need?
verwirrend gewesen,
Can it
weil wir da
put
einen
it on a
Tisch drunter
simple
gesetzt haben.
lab
Und der
desk?
ist ungefähr 70
It's
Zentimeter
about
tief,
70
der Roboter,
centimeters in
und
depth,
einen
this
Meter breit.
robot,
Also es ist
and a meter
vergleichbar mit so einem Eppendorf
broad.
Liquid Handler, vielleicht ein kleines Tick
Compared to an
größer.
Eppendorf liquid handler, it's slightly bigger in
Dann
its
noch mit die
dimensions.
Protokolle.
Well, a question about the
Standardisierten
protocols,
Protokolle,
the
die ihr auch
standardized
anbietet,
protocols you offer.
habt ihr die übernommen und
Have you
verfeinert
taken
oder
them over
implementiert ihr die
and
selbst?
made more sophisticated,
Aktuell ist
or
alles
do you set
übernommen
them
und
up?
verfeinert.
Currently, we have
Ist eine
taken
reine
over all of
Ressourcenfrage
them.
bei
It's
uns. Wir
and has
hatten
been
bisher halt nur
refined,
Julia, die
made
wirklich
more
im Labor daran
sophisticated.
arbeiten
We
konnte.
have
Das ändert sich jetzt
our
aber.
people working on the
Beim
lab,
UKE
but
haben wir
this is
eigene
changed. And at
Laborflächen, stellen Leute
UKA,
ein, zum eben auch
the
selbst weiter
University
in
Hospital of
Protokollentwicklung
Hamburg, we hire
einsteigen.
people
Wir haben so
that
ein paar Quick
enter
Wins schon
our
gesehen, wo wir
work
wahrscheinlich auch
from
viele
the
bestehende
university
Protokolle
side, and we have
optimieren
some
können.
quick wins where I
Werden da
was
aber
able to
jetzt deutlich
optimize
tiefer
some
bald
existing
einsteigen.
protocols, but we will take a more in-depth approach
Ja,
soon.
dann